Hi everyone, I am Dr. Anisha, your microbiology faculty, and today I will be discussing about dermatophytes. The dermatophytes, I will be covering up the different types of dermatophytes, their clinical types which are very frequently asked in your questions along with the images, or rather we say they will be asked as clinical scenarios and will be given images along with it. And the lab diagnosis, the differentiation or identification of dermatophytes on the basis of macroconedias as well as microconidias, right? And special tests which are also devised to identify these dermatophytes. Firstly, let us talk about what these dermatophytes stand for. So these dermatophytes, these are nothing but group of fungi which are responsible for superficial cutaneous fungal infections, and their common name, which most of you might have seen or heard in various advertisements, right? So these are called as Tinea infection or ringworm. So annular lesions on the skin are seen, and majority of the lesions are annular with active margins, right? So you might have seen this ring-guard ad in your televisions. This ring-guard is used for nothing but the tinea infections. So what are these dermatophytes affecting? These affect keratinized tissues. So naturally these are keratinophilic, right? And what are these keratinized tissues? These are nothing but skin, hair, and nails. So can we say that these dermatophytes are affecting skin and its appendages, right? So what are these appendages? Hair and nails. So in order to remember or to memorize what structure is involved in which type of dermatophyte infection, I have learned it this way, so it makes it very easy for you. Firstly, we have the skin, right? Secondly, can you appreciate these small minute hair follicles which are present on the skin? Hair follicles, right? And then on the upper portion, on the upper part of the finger, can you see only the skin and nail? So it is not just the finger which is getting affected, but the different appendages. And in order to remember what all are involved, you just need to remember this image of the finger, right? Firstly, we'll use this as a picnomic. Now talking about different types of dermatophytes, these are three: Trichophyton, Microsporum and Epidermophyte. So whenever we talk about dermatophytes, we have three types of dermatophytes: Tricho, Microsporum and Epidermophyton. So what are these different dermatophytes involving, right? So when we say Trichophyton, it stands for 3. See? So now all these three structures, that is skin along with the appendages, are getting involved. All three: skin, hair and nails. They get involved in Trichophyton infections. So when we said, let's refer to this image again. When we said Microsporum, so two structures are getting involved. So hair follicle along with the skin. So we have minute hair follicles present on the skin in this portion of the finger, right? So in Microsporum, two structures are getting involved: minute hair and skin. It is the hair and skin which are getting involved in Microsporum species or Microsporum type of dermatophytes. And then we have Epidermophyton. Epi stands for anything which is above. And what do we have above on the above section of the finger? We have just the nail along with the skin, right? So in Epidermophyton type of infection, Epidermophyton dermatophytes, they are associated or they will be involving skin as well as nails, right? Two structures are again getting affected or involved in Epidermophyton infections. Now, talking about the habitat of these dermatophytes, we must know that there are certain species which are exclusively associated with human beings, and whereas there are some species which are associated with animals and they might affect humans as well. And some species, some dermatophytes, they are found in the soil and they very well can cause human infections, right? So when we first speak about the habitat, we have three types of dermatophytes or three habitats. One are arthropophilic, then we have geophilic, and then we have zoophilic. Arthros will take the very first letter, that is A. A stands for Admi in Hindi, right? And Admi in English are men, right? So arthropophilic, these type of dermatophytes, they will be exclusively associated with human beings and they will be responsible for exclusive human infections, right? So men are, according to them, they are supposedly mild, right? So arthropophilic infections are mild, whereas they will be chronic in nature. The infections with arthropophilic species of dermatophytes, they are going to be mild. Mild infections would be present, whereas they would be associated with the humans for a longer period of time, right? So they will be chronically associated and they lead to chronic infections. So now when we talk about geophilic, geo in Hindi stands for Zameen, right? We can refer to this as Zameen. And what is Zameen? On Zameen, what do we have? We have soil, right? So these species are associated with soil. They can be isolated from soil, right? So geophilic stands for Zameen or species which are associated with soil or their common habitat is soil. And zoo, we can very well make out, zoo stands for the species which are associated with animals, right? So these geo and zoophilic species, they will be responsible for acute and severely inflammatory reactions. Inflammatory infections or reactions, and they will come early, they will also resolve early. So you must remember the kind of infections that these are associated with and what are their common habitats, right? Now talking about the most common species or the species which we need to identify, the type of dermatophytes we need to identify which are associated with humans, right? So I have a mnemonic for arthropophilic, right? So it says tabla master plays red flute and violin. So what are arthropophilic species or which are exclusively associated with human beings? So firstly we will have Trichophyton tonsurans, T40. Then we have Trichophyton mentagrophytes. Another species associated, it is Trichophyton rubrum. Rubrum stands for red. Then we have F. F stands for Epidermophyton floccosum, and V stands for Trichophyton violacum. Right? So these are arthropophilic species or exclusively associated with human beings. Then for geophilic, geo stands for Zameen. So we can take G as the letter, right? From G, geophilic only will derive the species. It is Microsporum gypsum. Microsporum gypsum. This is a geophilic species, right? So we have taken G for G. And zoo, so we have two animals and two species which are most common. One is Microsporum canis. Canis in general term stands for dog, whereas there's another species which is Microsporum equinus, which is a common word. Equine stands for horses, right? So we have zoophilic species, two most important species which are Microsporum canis and Microsporum equinus, right? So with the mnemonic and from the name itself, we have derived different species and which very well get asked in your exams. Now, in order to identify dermatophytes, we must remember we firstly need a sample. So what are the samples which we require? Samples are of course going to be the keratophynic samples, that is skin, hair and nails. Skin, hair, nail, right? So firstly, in order to get a skin sample, what all can we do? We either can have skin scrapings or we can take skin biopsies. But the most recommended sample for skin, or the kind of sample which we require to identify the dermatophyte, it is going to be the skin scrapings. And how do we do these skin scrapings, right? So can you appreciate this image? In this image, we have an annular lesion which is present, and can you also see these active margins, these raised margins, and somewhat clear centers? These are nothing but tinea infections. So what are we supposed to get from this particular site or this particular, and what kind of sample should we require? We need skin scraping. And how are we going to do skin scraping? We'll start from the center. And we will move towards the outer surface, or we'll move towards the margin of the lesion, and we will be crossing this margin, this edge, or these elevated sections. So we'll start from the center, we'll cross the edge, and we'll move towards the outer side. So this is how we take the skin scrapings from such kind of lesions. So the person in this particular image, you have a slide, right? And the person is taking the sample, he's scraping the skin, right? So one sample is just skin scraping, or in certain situations where you do not get skin scrapings, we can also prefer skin biopsies, right? So firstly, we need skin scrapings, right? And then for nail, we require nail clippings. Nail clippings from the active margins of the lesions. Active margins, or where the nail portion is affected with the different types of dermatophytes. And for hair, we must remember, you should remember these words, clippings or skin scrapings, right? And for hair, we need to pluck hair, pluck the hair, right? We require plucked hair. Because whole of the hair is affected, in certain types of invasions, or types of invasions of dermatophytes. We have ectothrics, endothrix. So we need to get into the root of the hair in order to find what kind of dermatophyte infection is there. So we need pluckings of hair. So after we get these samples, what we need to do? We first need to prepare a KOH mount, direct KOH mount. So what is this potassium hydroxide doing? This KOH is going to dissolve dissolve the proteins as well as the tissue debris, right? And after dissolution, it would be very easy for us to identify the type of dermatophyte, right? So most common concentration of KOH which we require in order to dissolve this proteins part or this tissue debris, it is 10%, and it is also a question, right? So what does this direct KOH mount show? It shows us hyaline, septate hyphae along with arthrochonidia. Arthrochonidia are nothing but the spores which are formed due to the segmentation of pre-existing hyphae. So the dermatophytes, the septa would always be, the hyphae would always be septate, right? And we can also appreciate these arthrochonidia. These are nothing but these box-like spores which are formed due to the segmentation of pre-existing hyphae, right? So direct KOH mount shows these hyaline septate hyphae, and this will somehow give us a direction that yes, dermatophyte infection is present, right? And what do we need to do next? We need to do the culture of these sample, right? So for culture, we would be requiring a culture media. So most common culture media used in fungal growth, it is 𝗕𝗵_𝗙𝗚, right? So on SDA the colonies would be formed, and which will give us the clues of macro as well as the microconidias. Macro as well as the microconidias, right? And these macroconidia, different types or shapes of macro and microconidias will help us identify the dermatophytes, the types of dermatophytes. Right? Now let us see what these micro and macroconidias look like. So can you appreciate this septate hyphae? This is one hypha, right? So there is septation present. So the smaller spherical structure which is derived from the hyphae itself, it is the microconidia. So micro and macroconidias, you must remember, these are asexual spores. Right? So these microconidias are naturally micro, so these are smaller in size, right? Smaller in size, single-celled, right? And these can also vary in their shapes. A question which gets asked is about the teardrop-shaped microconidia, which is a key specific feature of a species of Trichophyton, that is Trichophyton rubrum. It gives teardrop-shaped microconidia. It has teardrop-shaped microconidia, and this Trichophyton rubrum stands for red. So this imparts the color to the media or the colony. The colonies are red colored. So the media turns red and it gives a very beautiful color, Trichophyton rubrum, right? So microconidias are going to be single celled, they are smaller in size, they can be present in, their shapes can vary, and there can be few microconidias, there can be abundant microconidias. We'll see that in later section. So this larger structure, this is nothing but the macroconidia. Macro means large, right? And these are also segmented. You must remember, not just segmented. These are multiple cells. There are multiple cells present inside, right? So these are multiple celled, multicellular, multiple celled, and they have these walls around them. And for differentiation, you must know there can be smooth walled, and there also can have small granules which makes them rough. So these also can vary in their shapes, right? Varied shapes are present. These are larger in size and these are multiple celled, right? So this is the macroconidia, whereas this is the microconidia. Now talking about the differentiation of different dermatophytes on the basis of shapes and the number or quantity of macro and microconidias. So firstly, we will be discussing about the first dermatophyte, that is Trichophyton. So from Trichophyton, we will take the letter P, we'll highlight this letter P, and P stands for pencil. Remember, pencil for P, right? So these pencil shaped Trichophyton are going to be pencil shaped, right? Of course, they are going to be thin. They will have thin walls. They will have again smooth walls. And the ends are going to be blunted, blunt ends. And these macroconidia, these are not going to be very large in quantity, but these would be present in very less number. So these are less in number, pencil shaped, thin walled, smooth walled, and they'll have blunted ends, just like an unsharpened pencil. Whereas the microconidia in Trichophyton, see, P say also remember pack of pencil. So when we hold a pack of pencil and we put that on a piece of paper, so multiple black dots would be present. Multiple dots would be given by a pack of pencils, like abundant dots. So this shows that microconidias would be abundantly present for Trichophyton. And also these microconidias can be present singly along the hyphae, or they can also present as bunch or just like grape-like branches. So these can present as bunches or grape-like clusters, right? So now moving on to the next dermatophyte, which is Microsporum. For Microsporum, we'll take the letter S. We'll highlight this letter S. S stands for spindle-shaped macroconidia, right? It is spindle-shaped. It will have, naturally it is going to have pointed ends because it is spindle-shaped, right? Then the walls are going to be thick as well as rough walls, because a lot of granules, I have also seen a lot of granules present in the walls of these Microsporum, and these are very easily identifiable under microscopy, right? So these are going to be thick walled, rough in texture, pointed ends, and spindle shaped, right? So these are the features you must remember for Microsporum macroconidia. And what about the microconidia? Again, remember the letter S. S also stands for sparse, right? So the microconidias would be present, but these are going to be very less in quantity. They might be present in very less amount. So S stands for sparse also. So remember S for sparse for microconidia of Microsporum, right? And then let's move on to the third type of dermatophyte, that is the Epidermophyton. For Epidermophyton, I have a mnemonic which is 𝗖𝗵𝗵𝗀. Now, derma club, firstly club. Club stands for the macroconidia, which is the shape of the macroconidia, club-shaped. This takes up the first or some portion of the mnemonic, right? And the elite derma clubs, these are not meant for normal population, so very less amount of people would be there, selective people would be there, right? So club would be present, macroconidia would be present, and these are going to be club shaped, whereas microconidia would be absent, right? So elite clubs, these are not for normal population, so the microconidias would be absent. So remember, elite derma club for club shaped, and elite for no or absence of any microconidias, right? So what are these epidermophytons affecting? Epi means above, and above on the finger we had skin and nail, right? Now moving on to the identification of these conidias, right? So let's see these images and we'll be very easily able to make up the different types of dermatophytes from these images itself. So firstly, can we appreciate this elongated pencil-like structure? And these are also thin-walled, the walls are smooth, and we have blunted ends. Blunted ends are present. So these are nothing but this structure is like pencil. So this is the macroconidia of Trichophyton. Trichophyton macroconidia, pencil shaped. And also, can you appreciate these blurred out structures, some small structures which are present in bunches? I think all of us are able to appreciate these. These are nothing but the microconidia, and these are present as bunches or as clusters, right? So these are Trichophyton macro as well as microconidia. So this enlarged image of the microconidia, this is an enlarged image, right? So can we see these small spherical structures, round structures? These are nothing but the microconidia which are present as bunches. This is also an image for Trichophyton, right? Now moving on to the next image. I think now we are able to identify it very easily. It is spindle shaped with pointed ends, right? And if we look closely, these walls are not just smooth. We can appreciate the small granular structures which are present around. Small granular structures are present, and look at the thickness of the walls. Can you appreciate the thickness of the walls? These are thick walled, right? So this makes us identify the next dermatophyte, which is the Microsporum, right? And it is spindle shaped with rough walls. And in this particular image, can we also appreciate the small, if not many, quantity of conidias, right? So these are microconidias which are present sparsely, right? This is Microsporum. And very easy to identify now, it is the third type of dermatophyte, which is the typical club shape. A typical club shape of the macroconidia, and no granulation, no roughness is present in the walls. Walls are absolutely smooth, right? Thin walled, club shaped. These are nothing but Epidermophyton macroconidia. Now in this particular image, can we appreciate that those microconidias along the hyphae or anywhere, these are absent. So there are no microconidia. No microconidia, right? So this makes us identify, these images make us identify the different types of, the dermatophytes. these images, we can very well solve these image-based questions from dermatophytes. Now moving on to the different clinical types of dermatophytes, and firstly Tinea capitis, which is again a very favorite question among examiners. Tinea capitis. Tinea capitis, it naturally is affecting head and scalp, right? Cap stands, we wear the cap over the head. Capitis for head, right? So Tinea capitis can cause two types of infections, right? So these can be inflammatory Tinea capitis as well as non-inflammatory infections. Non-inflammatory infections. So non-inflammatory reactions include firstly black dot tinea infection, and the other type of infection associated with non-inflammatory tinea, it is the seborrheic dermatitis like and will have grey patches which will be present on the scalp. So no inflammation would be seen in the lesions or in or around the lesions, right? Whereas in inflammatory lesions, inflammatory tinea infection, there will be two types of Tinea capitis infection. One is 𝗘𝗅𝗙𝗏, the other one is 𝗕𝗵𝄔. 𝗘𝗅𝗙𝗏 as well as 𝗕𝗵𝄔. So firstly, let us talk about 𝗘𝗅𝗙𝗏, right? So 𝗘𝗅𝗙𝗏, it is nothing but boggy swelling which is well circumscribed, right? So let's first see the image for 𝗘𝗅𝗙𝗏. So this is a 𝗘𝗅𝗙𝗏 infection. It is well circumscribed, somewhat boggy, and lot of inflammation is present. Can you see this pus points, these pustules which are present, right? A lot of inflammation is present at this particular site. And can you also appreciate the alopecia here, loss of hair which is present, right? So in 𝗘𝗅𝗙𝗏, there is going to be a boggy swelling which is well circumscribed, and there's going to be a lot of inflammatory reaction. It will have pustules, and the hair follicles can be seen with discharging pus. Now, how do we remember 𝗘𝗅𝗙𝗏? So most of you might have seen Tom and Jerry cartoon, right? So you must also be remembering the kind of mischief these guys used to do, and how dangerous these guys used to get involved, dangerous fights these guys used to get involved in, right? And once Jerry used to hit Tom or vice versa, there used to be a small inflammatory lesion which would appear on their heads, right? Red swellings which would appear eventually after getting hit, right? So this is the inflammation, this is somewhat elevated. And so Jerry and Tom, these guys were both mischievous, right? So I have, for the species, I have a mnemonic which is derived from this particular cartoon, that Tom is a very mighty kid, right? So with this kind of lesion, we must remember 𝗘𝗅𝗏 is this kind of inflammation. Not exact inflammation, but it somewhat gives this picture, elevated, well circumscribed inflammatory lesion which is present, right? So firstly, we must know the type of alopecia. There is of course going to be hair loss, and this hair loss, it is going to be permanent in nature. So no hair would be grown thereafter, and the hair follicles will be destroyed. Hair follicles get destroyed, and they would be eventually they will be replaced by scar tissue. Scar tissue or crusts would be present, right? So permanent hair loss. So this kind of hair alopecia, it is known as scarring alopecia. Right? So now moving on to the most common species which are associated. So T stands for Trichophyton. Trichophyton. Right? And with V will take out the first species, which is Violaceum. And with M will take out the another species which is associated. It is Trichophyton mentagrophytes. And K stands for Cheiron. So in Cheiron infection, the two most common species associated are Trichophyton mentagrophyte and Trichophyton violaceum. Right? So this helps us understand what exactly Cheiron is. It is going to be an inflammatory form of Tinea capitis infection. So now moving on to another type of infection or another type of inflammatory Tinea capitis infection, it is the 𝗕𝗵𝗄s. 𝗕𝗵𝗄s forms cup-like crusts on the scalp, right? These are also going to be well demarcated. And these cup-like crusts are also known as scutellum, which is nothing but the shields which form around the hair follicle. Hair follicles. So these are also going to be elevated, enlarged. These are going to be very thick shields which are well demarcated, which would be present around the hair follicles. This shield is known as the scutellum, which is also a potential question. And let us see what this 𝗕𝗵𝗄s looks like. This is the type of crust which is formed over the scalp area. Cup-like crust, very thick in their nature, and they form somewhat honeycomb-like appearance. Honeycomb-like appearance. And of course, this is also a type of inflammatory reaction. So the alopecia, again, it is going to be scarring type of alopecia. Scarring alopecia would be present. And there's going to be a permanent kind of hair loss. Permanent hair loss. Right? And what is the most common species associated with 𝗕𝗵𝗄s kind of infection or Tinea capitis infection? Let's remember the letter S. S, we now know that it stands for shield, right? It stands for shield. It also stands for scutellum, right? So what is the most common species associated? It is going to be Trichophyton schoenleini. Schoenleini, it is going to be associated with 𝗕𝗵𝗄s infection, right? So this is also asked very frequently, what is the most common species associated with 𝗕𝗵𝗄s? It is Trichophyton schoenleini. Now talking about the non-inflammatory type of Tinea capitis infection, this is the black dot. In this image, in this particular image, can we see these small black dots? These are nothing but the broken hair follicles. Broken hair follicles, they are not uprooted, but these are broken because of excessive conidia proliferation, or the hair shaft, it is invaginated, or it has a lot of conidia invagination which is occurring because of the excessive growth of these conidias, right? Excessive conidia invasion, we can say. And so the hair follicle, it doesn't uproot but it breaks. and this gives an appearance of black dot, right? So alopecia would be present, of course. There's not going to be scarring type of alopecia. And what are the most common species associated? Remember TB, T for Trichophyton tonsurans and Trichophyton violaceum. So these are again the most common species associated with black dot type of alopecia or black dot type of tinea capitis infection, right? So this shows different types of tinea capitis inflammatory and non-inflammatory infections. Now what are the other types of tinea infections? Pedis stands for the infection of the web of the toe fingers or web of the fingers, right? Firstly, it is going to involve the web, and then it is going to involve the sole of the feet, sole of the foot in moccasin fashion. So firstly, it is affecting the web spaces and then the whole sole. The sole is getting affected. And what is this type of infection called? This is nothing but athlete's foot. A question, image like this would be given. You would be given a specific history, and this is going to be web spaces can be used as a buzzword for you. And this is known as athlete's foot, and tinea pedis stands for the infection of the web spaces, right? Cruris stands for the calf area, but cruris specifically stands for groin. We use cruris for groin involvement in tinea infection, right? So groin is getting infected, right? And this kind of tinea infection of the groin area, it is also known as jock itch. Jock itch. Groin area infection caused by these Teneas or Trichophytons or dermatophytes, it is known as the jock itch. Then Tinea barbae. Barbae, it stands for bearded area infection. Bearded area, it is getting affected in males, right? So in this image we can appreciate this inflammatory reaction. It is nothing but the tinea infection of the bearded area. Now Tinea manguum, it is the infection of the nail bed. Nail bed. So can you appreciate this image? Nail bed which is affected, right? So this is Tinea anguum infection and this is nail bed infection. Another infection involving the trunk, it is caused by Tinea corporis, right? Tinea corporis. A question which is now getting asked, it is the Tinea imbricata type of infection, which is there are going to be concentric lesions which would be present on the skin surface, right? Concentric lesions would be present, multiple concentric lesions which are caused by Tinea concentricum. And this type of infection, it is nothing but Tinea imbricata. Imbricata. This is tinea capitis infection. Another important two things which get asked, these are 𝗕𝗵𝗵𝗂, which is involving the non-bearded area. This is involving the non-bearded area, and Tinea manum, which is involving the Palmar aspect of hand. Palmar aspects of hand. So these also you need to know. So the most common species otherwise which are associated with these different forms, the most common remains the Tinea mentagrophytes or Tinea rubrum. So these are most common species, but the other species which we discussed separately for 𝗘𝗵𝗔 infections or for different infections, these can also vary. But the most common species or the type of dermatophyte which is associated with infection, it is Trichophyton infections, right? So now moving on to another type of granulomatous inflammatory reaction, which is a very hot topic nowadays. It is Mycology granuloma. So what is this kind of granuloma? So can you see this well demarcated lesion which has multiple nodules, right? So there can be multiple papules and nodules, and this is well demarcated, well circumscribed. So what kind of granuloma it is forming? These are well circumscribed granulomatous lesions, this raised lesions which are formed include various nodules, papules, right? And these are formed following any sort of trauma, right? So this is commonly one of an important point, that this can be formed in certain females who have the tendency to shave, right? But these can be associated with some form of trauma initially, which later forms into these papular nodular lesions, which involve the skin as well as the subcutaneous tissues, right? So these can be present over the legs, these can involve the face also, can involve the hands, etc., right? And this kind of granuloma, it can be present in normal individuals, whereas in certain immunocompromised individuals, these papules and nodules are going to get converted into deeper abscesses and pustules, right? So there will be deeper abscesses and pustules, which are also known as Mycology granulomas, but that too in immunocompromised individuals, whereas in normal individuals, so these will be associated with nodular raised granulomatous lesions, right? In normal individuals, these can be present and these are associated with trauma initially, right? So this is a question, Mycology granuloma, which gets asked from dermatophytes. Now moving on to a very important section where you get a lot of one-liners from. So the types of hair invasions. So we have, this is the structure of the hair. We have the outer part, that is the cuticle. Then we have the cortex and the medulla, right? So hair shaft is getting involved in certain dermatophyte infections. So what are the different types of invasions? There can be ectothrix-like invasion and there can be endothrix-like invasion. So what are these small structures? These are nothing but the conidia. These are nothing but the arthroconidia, which are growing inside or along the hair shaft. So let's see. Endo means inside, right? So the conidia invasion, it would be present inside the hair shaft. The conidia will grow inside the hair shaft, making the hair weak, and also it damages the cuticle as well. So conidia invade the hair shaft So they grow inside and ultimately leading to damage of the cuticle. This is one type of invasion, endothrix type of hair invasion. And then we have another type of invasion, which is the ectothrix. Ecto, from the name itself, we can make out that this stands for anything which is outside, right? So these conidias would be present outside either as mosaic fashion, or these can be present in chains. Can we appreciate these small chains, right? So these can spread as mosaic fashion or as chains outside the hair shaft, along the hair shaft, and naturally it is not going to invade. So the cuticle, it is spared. Cuticle is spared in ectothrix kind of invasion of the hair shaft. So these are two very important sections or points which you need to remember from this type of invasion, or the invasion which is done by dermatophytes. So now what are the examples of these invasions? Ectothrix as well as the endothrix. So I have two mnemonics for these again. So my cat gets very red apples. So let's see what are the species which have ectothrix kind of invasion and the species which have endothrix kind of invasion. M stands for Trichophyton mentagrophytes. C stands for Microsporum canis. Then G stands for Microsporum gypsum, which we have already seen under the 𝗘𝗵𝗃𝗀 habitat, right? So G stands for gypsum. Then we have V, it stands for Trichophyton verrucosum. R we have already discussed, Trichophyton rubrum. And A stands for, it is Microsporum epidermophyton. Microsporum epidermophyton. This is also an important species, right? So these are different ectothrix invasions, and the endothrix kind of invasion, it is shown by three species. Firstly, end stands for endo, right? S stands for Trichophyton 𝗪𝗪𝗓𝗍𝗓𝗔𝗍𝗔, right? Then T stands for Trichophyton tonsurans, and T also this V stands for Trichophyton violaceum. So these are the three species which would show endothrix-like invasion. And one important point which I forgot to mention in one of the sections, it is the 𝗕𝗵𝗆. What is the most common species associated with 𝗕𝗵𝗆 infection? S, okay, I told that already, Trichophyton 𝗪𝗪𝗓𝗍𝗪𝗓. So this was about the examples of ectothrix and endothrix, right? So now moving on to the lab diagnosis. Firstly, samples. Some of the sections we have already seen, but little bit more detailing would be done. So for sample, we have seen we require skin scrapings, hair, plucked hair and nail clippings. So before scraping the skin, we must remember that we need to disinfect the skin with alcohol, right? First we need to disinfect with alcohol, and then we take the skin scrapings from center to edge. center to periphery, right? And these nail clippings are supposed to be kept in the brown papers. Brown papers, and they're kept folded. Kept in folded brown papers or black papers, right? So now for direct KOH mount, we said we require a particular concentration, right? So 10% of KOH is the most common concentration that we require, but in certain harder structures, certain structures which are hard to dissolve, so we require a more contact. Firstly, we require a longer contact period and then higher concentration. So what we do for say for nail samples which are harder to dissolve, and also for skin biopsies, what we do, we increase the concentration of KOH. So up to what concentration of KOH can we increase in order to dissolve the proteins, right? We can increase the concentration to 40% for hair as well as biopsy samples. Not hair, it is for nails. So for fungal culture, we have seen it is done on SDA culture media, which is help us going to detect the macro as well as microconidias, right? We are doing fungal culture. Another thing that we do for identification of this particular another test which we do for identification of different these things for identification of dermatophytes, it is one thing which is DTM media, which is nothing but dermatophyte test media, dermatophyte test medium, which is specifically used for identification of dermatophyte. So what do we do in this type of identification test? That this particular media has a specific indicator which is phenol red. So what do these dermatophytes do? They are going to, because of the metabolic activities, in around 3 to 6 days, these dermatophytes are going to impart the pigment to the phenol red. So the medium turns red because of the metabolic activities of dermatophytes, right? And what so this helps us in presumptive identification of dermatophytes, dermatophyte test medium. So the medium turns red in around 3 to 6 days, which is a specific feature of certain dermatophytes, which is again helping us in presumptive identification of the dermatophytes, right? Then for Wood's lamp examination, we must know that there are certain dermatophytes which fluoresce when viewed under the Wood's lamp, right? So why this fluorescence is occurring? It is because of the presence of a pigment which is pteridine pigment. Pteridine pigment is present which in the cell wall, which is helping us identify the fluorescence which is imparted because of certain which is imparted in certain dermatophyte species. And what are these dermatophyte species which are going to fluoresce in after examining under Wood's lamp examination? These are two species. One is Trichophyton mentagrophytes and Trichophyton 𝗘𝗵𝗓𝗏. Right? So this is the mnemonic for Wood's lamp examination. So bright green would be given by Trichophyton mentagrophytes, whereas dull green fluorescence would be given by Trichophyton 𝗘𝗵𝗓𝗏. Right? And the fluorescence is because of the pteridine pigment. So we detect fluorescence in dermatophytes Fungal culture we are doing on SDA media and the dermatophyte test medium. Dermatophyte test medium. So the color imparted to the medium would be red because of the metabolic activities of dermatophytes. Right? So now let us also know what is this hair perforation test, which is also an important test which is done for identifying different species or to differentiating between different species of dermatophytes. So this is a test specifically done to differentiate between Trichophyton mentagrophytes versus Trichophyton tonsurans. One thing. And two other species for Microsporum, these are also used to differentiate for the presence of this hair perforation test. So these are Microsporum canis versus Microsporum equinus. So for hair perforation test, let us just look at the image. Can we see these small wedge-like invaginations which are occurring? This is the hair shaft, and these are wedge-like invaginations which form inside the hair shaft. So if this forms wedge-like invaginations, if they form, this gives a hair perforation test positive. Test positive, right? And what are the species which are giving this test positive? So the species between Trichophyton mentagrophytes and Trichophyton tonsurans, Trichophyton mentagrophytes, it is going to give a test positive, and this is going to give a negative hair perforation test. And between Microsporum canis and equinus, canis is going to give a hair perforation test as positive, whereas equinus is not going to give a hair perforation test. It is not going to demonstrate this wedge-like invaginations, right? So this is a test which is going to differentiate between these four species, right? 2 and 2. And then another test, urease test. So one species, Trichophyton mentagrophytes, it is going to hydrolyze urea, which is going to convert the color of the medium, urease medium, to bright pink. And so this is the only species which is going to give urease test positive, which is also a one-liner question for your exam. So another test which we can do for identification of genes, it is the PCR. We are going to identify the chitin synthase gene for dermatophyte identification. So this was about the important lab diagnostic tests which are done, right? And different samples which we use, different tests which can be done, and specific tests which are done for dermatophyte infections. So this was all about the high yield points from dermatophytes. Thank you.